Welcome to Mystery Biology. Let's learn something new. Here in this video we will talk about alpha complementation and blue-white screening. Here we will also see the role of beta-galactosidase enzyme and how it converts X-gal into blue color product. We will also see the role of insertional inactivation and how it is related here with this alpha complementation. So let's move on to the topic. First, let's talk about beta-galactosidase gene. Here this beta-galactosidase gene, or we can say this Lactose gene, it is a structural gene of lactoferrin which is present here in this bacterial chromosome. Okay, and this Lactose gene produces the beta-galactosidase enzyme. And this enzyme converts lactose into glucose plus galactose. Right? And here this beta-galactosidase is a polypeptide of 1029 amino acids, and this forms after tetramerization. So we can see the step tetramerization is very important here, because after this tetramerization the active form of beta-galactosidase enzyme produces. Okay, and this tetramerization is again dependent upon the presence of N-terminal region of this gene. We will understand this point in my next slide. Okay, suppose if the Lactose gene is normal, that means it can produce the active form of this beta-galactosidase, right? And suppose if we delete some part of this gene, suppose here we delete the N-terminal region of this beta-galactosidase gene, then it would not be able to produce this beta-galactosidase enzyme. Okay, so by doing this we are creating a mutant form of bacteria in which we are deleting some part of the gene, right? Now suppose this is beta-galactosidase gene which is present in lactoferrin, and here we delete the N-terminal sequence of this beta-galactosidase gene. Then after the deletion, the peptide which forms after the translation is called the omega peptide, and this omega peptide is unable to form the active form of the protein, right? Now we can see that this omega peptide is not able to produce the active form of the enzyme, right? And if we provide the N-terminal sequence through this vector, when the bacteria is transformed with a vector which contains the N-terminal sequence. Okay, and this N-terminal sequence then later translates, it forms the peptide, and that peptide we call the alpha peptide. Okay, so here this alpha peptide acts as a complement for this omega peptide, so that together they form the active form of enzyme. And that active form of enzyme can converts this X-gal into blue color product. Okay, so we can see the rescue of the function of mutant beta-galactosidase by the alpha peptide is called alpha complementation. Okay, now let's understand this more clearly. Suppose here we take the mutant strain named Lactose delta M15. This is the name of the mutant strain. Okay, and we have seen the bacterial chromosome. Okay, here this bacterial chromosome has the Lactose gene. Okay, and here this Lactose gene we have deleted the N-terminal region, and now it can only produce the omega peptide, right? And suppose this bacteria is transformed with a vector which has the N-terminal region, and here this N-terminal region, we can also say this Lactose alpha, because ultimately it is producing the alpha peptide. Okay, and now together this alpha peptide and omega peptide, when they express together, they form the active form of beta-galactosidase, which can now hydrolyze this X-gal, which is colorless, into blue color product, right? And in the second case, suppose if we insert our gene of interest but not in this portion, okay, not in this N-terminal sequence, we are inserting our gene of interest some other way, some outside of this Lactose alpha. Now what happens, since the gene is present some other way, that means our gene, this Lactose alpha is intact, and if this gene is intact, that means it can normally form the alpha peptide, and the rest you know very well, right? So if suppose gene of interest is not present at this portion, then again the result will be normal. That means active form of enzyme produced, and it can now convert this X-gal. Now here this vector carries multiple cloning site at this Lactose alpha region, and here we use the restriction enzyme to cleave at this portion, okay, so that we can add our gene of interest. And if suppose we are adding our gene of interest at this portion in this LAGZ alpha region, that means now we are inactivating this LAGZ alpha because of the insertion of our gene of interest, okay. So this LAGZ alpha, because it is now disrupted, it would not be able to produce the alpha peptide now. And if alpha peptide is not present here, then it would not be able to complement here this omega peptide. So no active form of enzyme can be produced and no XCal can be converted to blue color product. That means XCal will remain as colorless. So we can see because of the insertion of our gene of interest, now we are inactivating the original function of the previous gene. So we can see the role of insertional inactivation here, okay. So now we can see that the vector with foreign gene insert in LAGZ alpha does not form the active protein. No active protein means no blue color product will form, right? Since in this video we are talking about XCal again and again, so it is important for us to know a little bit about this. This is the structure of this XCal, and here this XCal, the full name is 5-bromo-4-chloro-3-indole beta-D-galactopyranoside, and it is a chromogenic substrate. That means it produces color, okay, and this is substrate for beta-galactosidase, which yields dark blue color after enzymatic activity. And also we know that this XCal is an analog of lactose. So suppose here we take the XCal in the agar plate and we also add the inducer IPTG for the lactoperon, okay. And now if suppose the active form of beta-galactosidase is present, then it cleaves. Let's see the chemical reaction here. If we use the beta-galactosidase enzyme here, then it cleaves this galactose unit. This is the galactose unit present here. It cleaves only the galactose unit from this whole XCal, okay. And now it forms the 5-bromo-4-chloro-indoxyl, okay. And now this compound spontaneously dimerize and oxidize with this protein, with this compound again, and now it forms the blue color product, which is 5,5-dibromo-4,4-dichloro-indigo, which is blue, okay. So this is the main chemical reaction which occurs here, right? So far we have seen how blue color colonies form, how white color colonies form, okay. So now let's talk about this blue-white screening. Suppose we take the mutant bacteria which is transformed here with a non-recombinant vector. Non-recombinant vector means that the LAGZ alpha gene is intact. That means no gene of interest is present at LAGZ alpha. LAGZ alpha now form the alpha peptide, which can again complement the omega peptide and then form the active protein, and that active protein can convert here this XCal into blue color product, okay. This is simple and you know it very well. And in the second case, suppose the mutant bacteria is transformed with a recombinant vector. Recombinant means here the gene of interest is present at the LAGZ alpha portion, right? So LAGZ alpha is disrupted, and since it is disrupted, it can now cannot complement the omega peptide, so it cannot form the active protein. And if no active beta-galactosidase is present, that means no XCal can be converted to blue color product, so it will remain as colorless, okay. And in the agar plate we can see this as white colony. So this is the plate, agar plate, okay. Now we can see if the bacteria is transformed with a recombinant vector, it now forms the white colonies, and if the bacteria is transformed with a non-recombinant vector, then it forms the blue color colonies. So now this is very clear to you, right? That was all about alpha complementation and blue-white screening. If you have any doubts, you can ask in the comment section below. And if you like my video, please like, subscribe and share with your friends. Thank you.